Hello, students. I wanted to do a quick overview of how you can use this photosynthesis simulation to see how different factors might impact the rate of photosynthesis. Over here on the left-hand side, you see all the factors that you can change. You can change whether or not it's in the dark or in the light. You can also change whether you have a specific spectrum of light or whether you're looking at full light. You can change the distance from light. You can change the initial pH of the stroma. You can add NADPH. You can change the initial concentration of carbon dioxide. You can change the temperature. And you can change whether or not you add this herbicide that actually influences the rate of photosynthesis. The first thing that we're going to look at is temperature. So in order to run this, I want to toggle this little guy here to 0, to start with 0 degrees Celsius. Then there's these different graphs here. I can look at glucose produced, ATP produced, NADPH produced, or oxygen available. All of these things are different ways that I can look at how fast photosynthesis is happening. I'm going to start by looking at oxygen available. Then, in order to run an experiment, I just click Run. I can see at 0 degrees, it looks like there's very little oxygen produced. We always want to run multiple trials. In this case, we'll run 5. Then if you go over to this graph and you click, then you can and hover over the 40-minute time, then you can see how much oxygen was produced. In this case, I can see it was 0. Then I can reset that and try it again. My next temperature is going to be with 10 degrees Celsius. So again, I can run that. I see there's a lot more oxygen available, meaning more oxygen has been produced through photosynthesis. Again, I can click and hover over the 40 minutes, and below it'll tell me my numbers. So 12.1% for my first trial, 12% for my second trial, 12.1% for my third trial, 11.8% for my fourth trial, and 12.1% for my fifth trial. I'm going to repeat that at 20, 30, 40, and 50 degrees so I can see how those factors of temperature influences how fast oxygen happens. You might choose to use this spreadsheet to put your data in. If you do, then this chunk over here, the average relative percent oxygen at different temperatures, will be what you graph. If we had time, then we could draw on error bars showing standard deviations. It's really hard to do in sheets though, so we'll probably skip that this time. Please remember when you're creating your graph, you want a really descriptive title, and both of your axes need to be labeled. One way you can do that is if you click these 3 little dots and go to Edit Chart, go to Customize, then on Chart and Axes Titles, you have this drop down where you can change the titles so they match your data in a really descriptive way. After you've done this for temperature and how it influences the rate of photosynthesis, you're going to choose your own variables. Do you want to change light? Do you want to change pH? Do you want to change NADPH, carbon dioxide, temperature, or the presence of this herbicide? You're going to design your own experiment to see how those things impact the rate of photosynthesis. One last thing, up top here, there's a second graph. This graph is showing all the different variables, all the possible dependent variables. I chose to focus on oxygen available, but you could focus on ATP, NADPH, glucose, or oxygen. You can click up here and you can see numbers for each one of those. That might help you make a decision when you're designing your own lab on what particular dependent variable you would like to record. If you have any questions at all, please let me know. Thanks.